So yeah, I'm Catherine, she, her, hers, and I'm a third year PhD student in a lab here at Rockefeller. And we study the bacteria that causes the disease tuberculosis. You may not have heard of it because it's not super big in the United States, but it's actually the #1 infectious killer. And when I'm not in lab, like I said, I like running. I have 2 cats, and just to try and get to know everyone, we have a little icebreaker about seasons. So let me pull up the poll. Yeah, so let me know what your favorite season is. I'll give you guys a couple seconds to fill that out. Okay, almost everyone. There's just like 3 people we're waiting for. Oh, we have people who have puppies in the chat. Awesome. I had a dog growing up, but now my apartment's too small. So we have 2 cats, Fifi and Ellie. They came with the names when we adopted them. So since this is just an icebreaker poll, I'll give you guys 5 more seconds. Okay. And yeah, there's a slight preference for winter. My favorite season is fall, so kind of sad that we're tailing into winter now. I like leaves and I like cinnamon, so. Oh, okay. We have love for summer. Oh, and a cat. So cute. Yeah, so I will. How do I do that? Sorry, I'm still getting used to Zoom. Okay. Yeah, so just stop me anytime if you have questions or something you want to see more or different. But you are in a microbiome class, and I study bacteria. So I wanted to give you a little bit of a tour of what our lab looks like. So you'll notice I have these purple gloves. I wrote on this one in Sharpie because we want to keep everything away from us. So I take them on and off when I go to touch my phone or touch my computer, just to keep everything clean. And so I'm going to take one off to grab my phone. But if I'm doing this correctly, and there's another window where you see like a blue rack with a bunch of tubes and stuff. So if you can see that, that's just a bunch of DNA, but looks cool. So it looks like it's just water, but there's actually that tube is empty. Let me get one that has stuff in it. Yeah, see at the bottom there, it looks like there's water, but that's actually a bunch of DNA. So super cool. But we're here to talk about bacteria. So I wanted to show you guys some different types. So there are bacteria that most people have heard of, like E. coli. Oh, sorry. You can spotlight 2 videos. That's so cool. So most people have heard of E. coli, either from like food things, or if you're in a bio class, you might have heard it in reference to like cloning or genetic engineering. But this is what E. coli looks like on a plate. You can see there are these little colonies that grow, those individual spots, and then this big section that we call a lawn or a smear. Can you see that okay? Nice. So there are lots of different types of bacteria. So there's E. coli that most people have heard of, and then I study what's called a mycobacteria, MYCO, and they look a little different. It's hard to see from this side, but before I open them, we have to turn on the flame. So I'm gonna try and put this up here. Oh, cool, you can still see it. Basically, oh, someone asked what the DNA is from. So I actually pulled it out of E. coli, so out of these cells. I grew them up in a liquid, and I put a bunch of chemicals in to break them open and pull out their DNA so I can figure out what it is, and then I can put it into other bacteria. Basically, I want to use something that the E. coli have in their DNA, and I want to express it or make it happen in a different bacteria, one of the mycobacteria. So someone's asked what I mean by a flame, and I'll show you. So when you work with bacteria, not only do you need to protect yourself from them, we wear the gloves, and if you work with a pathogen or a bacteria that causes a disease, you have to put on like a different kind of mask and special, like a lab coat. But we also want to make sure that the bacteria that we're working with are protected from other bacteria that might be on our hands or in our breath. So I take ethanol, just like hand sanitizer, but without the nice smells. And I wipe down my gloves and my area to kill all of the bacteria that are just hanging out on the surface. And then I turn on a legit flame. So I'm trying to get it within both cameras' views. Nice. And I have a little striker here. Make a little spark. Some gas comes out of the canister. And you can see that there's a flame. So this is called sterile technique. We use it so that we can protect the bacteria from bacteria that we don't want. So I want to make sure that everything on this plate is what I think it is, what I know it to be because I put it on there. So by using a flame, we can protect the bacteria from other things, just like we use gloves to protect ourselves. So bacteria look kind of different. And let me just take off a glove so I can show you that. So I can get it to focus. Sorry. Is that focusing okay? Or is it? Probably not super great, but you can see how they look kind of wet, kind of like gunky, like a grape if you've washed it. And then these ones look waxier. It's hard to tell on the camera, but there are also like little dots in the center. And that's because these two bacteria grow differently. Someone asked in the chat, is it true that the blue part of the flame is the hottest part? Yeah, so the blue and white flame are the hottest, and then the red and orange are cooler. And I think it also has to do with how much oxygen it's consuming, but it's been a while since I really investigated the flame. So I'm gonna leave you with the view of some very fun bacteria and ask a quick science question. Second. Oh, that's way too high up. I'll just hold it. Da-da-da. Cool. So let me just open a poll, one moment. So I mentioned this before. I wonder how many of you guys caught what a pathogen is. Great. Oh, very nice so far. Okay, there are 2 more people, but that might be the Rock EDU count and your teacher. So I'm gonna close the poll. Stop me if anyone didn't get to. Okay. Y'all are great. You're really killing it already. So yeah, a pathogen is a microorganism that can cause diseases in people. There are also pathogens that cause diseases in other animals, but usually when we say pathogen, we're talking about ourselves because humans are a little self-centered, you know how it is. So I'm gonna stop sharing the results. But real quick, some of you selected a microorganism that lives in animals and helps them digest food. So not all microbes are bad. Some of them are really good and help animals like cows digest foods. And those are commensal bacteria. The CO means you're working together, commensal, living together, helping each other. And we have those in our gut too that help us like digest food and make sure everything's working all right. And then there are pathogens, so bacteria that cause disease, like tuberculosis that I studied, or like someone brought up in the chat, the COVID-19 virus. That's another type of microorganism. It's a virus, not a bacteria, but that also causes disease in people. So while my lab doesn't study COVID, we share a higher safety level lab, and I'll explain that in a moment, with people that do study COVID. So there are different levels of safety depending on how hazardous the microbe is. So what we're doing here is called level 1. Basically, none of this can hurt you. If I wanted to, I could like put it on my hands and nothing would happen. Then there are level 2 where like, it wouldn't be great, but it probably won't get you sick, or we can like treat it really easily. And then COVID and tuberculosis are level 3, which means it's hard to treat or dangerous, and you can put in these external measures to make sure that you're safe. So I'm going to quickly share my screen, or, oh, I can upload a file to the chat. This is so cool. How can I do that? Can I just copy and paste this? Let's find out. It won't let me paste a photo. Okay, I'll share my screen super quick. So this is actually my lab in our safety level 3 lab. You'll notice that because we're working with a pathogen, a bacteria that can hurt you, we all have these special masks or these hoods on that filter air. We have these fancy overalls that are made of plastic to keep us safe. And we all have hair nets, so we look kind of silly, but it's one of the ways that we can be safe studying a bacteria that's harmful. I'm gonna stop sharing that, but you can see we have a lot of fun, so science is pretty cool. And we get to look like we're in a movie, so who doesn't want to feel like a movie star? But not all bacteria are harmful. you guys know that baker's yeast is a microorganism, something that's really small, but we use it to make sourdough and other types of breads and things. And there are different types of microorganisms, bacteria, viruses, a fungus, and then these other things called archaea. They're kind of like really old bacteria, but I don't know a lot about them, so don't ask me about that one. Real quick, I just want to get y'all's opinion, or would you mind answering this poll? Which of these is not a bacteria? Oh, forget about the last. Oh, actually, no, go ahead. Go ahead. Which is not a bacteria? There's more than one right answer, so you can put in either of them. And while you do that, someone in the chat asked if it's safer with the mask or the hood. Both are quite safe. Technically, the hood is a little bit safer, but we kind of just do it because it's more comfortable, usually. It's like a cool breeze from the back of your head. And someone says they have eczema. Me too, that's why I put it as one of the options. That may have given away whether or not it's a bacteria, but we'll see. Okay, everybody, 3 more people. I'm gonna give you 10 seconds. Y'all are rock stars. You're doing so great. Yeah, eczema is not a bacteria. It's a funny word for a special type of dry skin that's kind of itchy and red. Sometimes it can be quite bad, but most people, like myself and Joshua and Ethan in the chat, you just get it in the winter, little itchy. You put on some Aquaphor or some kind of cream and it feels better. E. coli is a bacteria. You'll remember that that was actually the first bacteria I showed you. So, one moment, I'm putting on my glove because I want to touch bacteria. Ooh, made a fart sound. So it's this guy. Oh, I just realized the person who's not responding to the poll is me. It's me from my phone. My bad. Yeah, so this is E. coli. Yeah. So it can look kind of different. This is what it looks like in a plate. That's pretty cool. It's like individual dots. And here I grew up a whole bunch of it. And then I spun it really, really quickly to separate out media, or like the good juice it likes to grow in, from the bacteria itself. So it's like a gross pellet. It's pretty cool though. Yeah, so that's E. coli, which is a bacteria. And then the other option was Salmonella. Salmonella is actually also a bacteria. I believe it's Salmonella enterica, just meaning that it's typically in like the human digestive system. So we've gone over eczema is not a bacteria, skin condition, and malaria is also not a bacteria. It looks like it might be because it has one of those good genus species, those Latin names next to it, Plasmodium falciparum, but it's actually a parasite. It's a type of cell called a eukaryote. I think you may have covered that in class, actually. where bacteria are prokaryotes and multicellular organisms like us are eukaryotes. I'm getting thumbs up, nice. Yeah, so malaria is a parasite and a eukaryote. So that one was pretty tricky, but you did really great with all of these. Nice. So I'll save the last poll for a little bit and just show you some of the cool stuff. This is like show and tell for me because I really like doing all this. So let me grab something from our big incubator. I'll bring you with me on my phone so you can get a good look at what the lab is like. Thank you for changing the view. So yeah, this is my whole bench. I apologize that it's messy. I was in the middle of stuff when you called. This is you. Creepy, huh? Yeah. This is the thing that spins really fast. You can put stuff in it and it'll separate it based on how dense it is. So if it's like really, really heavy or not that heavy. And then I'm gonna take you to the incubator. We have the tape so that we walk in only one direction for the COVID safety. So we grow a lot of bacteria, and part of that means we have to have a lot of incubators. This machine right here, it's an incubator. What it does is keep the bacteria at the temperature they like to be and spinning at the speed they like. So if we open the door, we'll see we have a bunch of bacteria. I wanted to show you this. This is a whole bunch of that mycobacteria, the MYCO I was talking about. You can see it kind of makes like a little gross film on the side. Let me see if I can get a better angle for it. Yeah, but it's like swirly and smooth. And I think it's really cool, but it's also kind of gross. So we have a whole bunch of those because everyone in our lab grows a lot of bacteria. So we need a lot of room for it. We also have incubators that don't move. So these are standing incubators. And instead of putting in tubes with that liquid like I showed you, we put in plates, those little Petri dishes that we looked at my bench. So you'll notice we have not too many plates in there right now, but it's just that we have so many. So we can put different plates in, different kind of things. This is one of the things that I don't know as much about, but I'm gonna learn more today. This is our microscope. It's super fancy. It has this special thing up here called a cell ASICS. I don't totally understand it, but basically we can put bacteria in a dish and then put that dish inside the microscope here. See that plate over there where I'm pointing? We can put bacteria in that, and that machine will give them the juice, the media they like to grow in, and we can watch it grow in real time on the computer. So give me one moment, enjoy our good mouse pad. But I know this is a janky way. Oh no, Windows. Remind me later. Let me find a cool video of bacteria growing. I know I could just share the screen, but I can't do that from this computer because I'm not in Zoom. So, come on. No, not Edge. Come on, Microsoft Edge. Can you see that? Isn't that wild? That's that mycobacteria I was talking about growing. So we take a picture every couple of minutes and you can watch it grow. See? Look, this one gets all curly because it's a mutant. I've made it so it can grow in a special antibiotic, something that kills it, but that means there's something wrong with it so that it grows all curly. And we don't know why that is, and it's so interesting, and that's what I'm gonna look at. So today I'm actually gonna put bacteria in the microscope that are red around the outside. It's red on the cell wall. You may have covered that in class too. So I can look at how their cell wall is growing, like if it's doing the right things or if something's messed up. And that's super cool, and I'm really excited to see what happens. But this is our giant microscope. I'm gonna take you back out to the rest of the lab. Yeah. So we have a good biohazard sign because in this other room, we have a BSL-2 hood. So you'll notice these are all closed and there's like glass. So what you do when you're working with something, a pathogen that can hurt you, you only work inside one of these hoods, and there's special air moving through this machine so that everything gets sucked inside and then decontaminated. Because if you just had it out in the air, it might like get on you and you might breathe it in. But this way, all the bacteria, all the pathogens stay inside, and we have our good lab coat that we wear when we're using this. And then we have special disinfectants, these cool pipettes. I'll show you those at my bench. And we keep everything really safe. We even have little instructions so that we don't forget. And it lets us study things that would otherwise might hurt us. I'm just gonna move back to my bench. So we have a lot of these cool hoods. Only that one gets the good bacteria in it. These ones are just to keep stuff clean. So we'll walk back. And I'm just checking the time. Oh, good. Have like a half hour, I think. So I have bunched up my gloves, so I'm just gonna grab a new pair. Put y'all back up here for a second and show you some of the cool tools we use. And if you have any questions like about this or about science in general, just drop them in the chat, unmute yourself, whatever you'd like. I'm happy to talk about anything. Maybe even about like where I went to school, how I ended up doing this. I'm an open book. But we have some pretty cool tools in lab. So can I, this? Yeah, this is called a pipette. Basically, it lets you suck up and dispense really, really small volumes. So let's do this one. We use what's called a microliter. So a liter, like the big things, like a liter of soda that you see at the grocery store. And then this is 10,000 times smaller. So itty bitty teeny tiny, and I'll show you that. We put on these little, like, we call them tips. They just go on top of this, and it lets you suck up just the amount you want. So you bang it on there, and you'll see that there's this little clear plastic tip. And so if I want to suck up a really small volume of something, let me grab something to use. This will work. And I'll try and show you up here. It's kind of hard to see, my camera's not great. You put it in, you press that tippy top part, suck it up slowly, and then way right there. You can't, see there's a little bubble there? I'll pull it up with the other camera as well. One moment. Gotta take off my glove. Will it focus is the question. Not really. I don't know if you can see that there's a little round drop of water right there. It's hard because it won't focus, maybe. But it's a super tiny amount, and it lets us make really, really accurate, so we know exactly how much is in that drop. And it helps us do reactions. So just to go through some of the questions in the chat, do I work on anything else? Let me actually cover some of these plates so that I don't waste flame while I'm here. So not right now. I used to, when you go to grad school, like I'm in, you do these things called rotations. Basically, you try out a couple different labs and things that you study to decide which one you're going to do a thesis in, a really big study of something. So I worked in a lab that studied viruses, a respiratory virus called RSV. Not a big deal. It's only like really a big deal if you're pregnant, but I only worked there for a little while, so I'm not super familiar with everything. And then I worked in a lab that studied how this protein, an enzyme, a protein that can do a reaction or change something into something else, how this enzyme that was made by the bacteria in our guts is able to keep us safe by fighting bad bacteria by cutting them up. So I did that for a little while, and that was really cool. And so that's what I've studied a little bit of. Oh, it's a Band-Aid on my left thumb. Sorry. The cats, you know, sometimes you pet them too much and then they scratch you. Yeah. Do you study a specific element here? So part of the research that I study, technically what I study is how bacteria that are able to survive in an antibiotic, a drug that would kill them, how they do that. So what are they using that helps them stay alive? So basically, it's like if you're already kind of sick, what things are keeping you from being any sicker? And I look at that in tuberculosis, the bacteria that causes the disease tuberculosis. But yeah, basically, how are these really dangerous pathogens able to survive? Someone asked, did I always want to do this when I was younger? I always liked science, but I didn't realize that I could be a scientist. Like, I didn't have this kind of thing when I was in school. So I didn't think like I could do it. Didn't realize like normal people became scientists. And then, so I went through high school and I had a really nice teacher who kind of told us like, oh, you can do this. And he encouraged us to like go to college for it. So I studied biology in college, biochemistry, basically just more chemistry in there. And then I ended up coming to graduate school here. So I'm from Long Island. I'm from Nassau County, the middle and the bottom, basically. And then I went to school on Long Island also. It's a little early for y'all to be thinking about college, but you can always think about that if you want. Where is my lab? So we're at Rockefeller University. So we're on the Upper East Side, and then we're in this open space building. So someone asked about Ebola. So I'm actually going to do a quick Google. So Ebola is a virus. Let's see what the CDC says, because they are great at this. So it's a virus, it's not a bacteria. So viruses can't multiply unless they're in a host cell. So in this case, the virus, that big piece of DNA, can't make more of itself unless it's inside a human cell. And that's what it does. It enters your body, it goes inside one of those cells, and it says, all right, stop what you're doing, make more of me. And then it makes more and more and more of that, and then the cell breaks open. And now each of those particles goes into another one of the human cells and says, stop what you're doing, make more of me. And that's the cycle in how it can get to different cells. Let me see. Yeah, it's mostly, it's like a blood-borne disease. So it'll be if you contact someone who's infected their blood or saliva, or you're bitten by like an infected animal. You might remember that was a previous pandemic. So I'll get right back to the RNA question, but no, Ebola can infect different types of organisms. So here, it can infect fruit bats and non-human primates, basically apes and monkeys, that kind of thing. So not just people, just in my example, I was saying people. Why does this sound like RNA duplication? So that's a super good point. Yes, Ethan brought that up. So viruses are just pieces of DNA or RNA. And so the way RNA kind of gets turned into a protein and you make many, many copies of that protein, you're just making more and more virus. It's the same way you like go from DNA to RNA to a protein, but in this case, just make a whole bunch of that genetic material. Does that make sense? Cool. I'm trying to think if there's anything else I have cool that's out. I don't know. Let's see what else I got. So I put on my gloves that way where I kind of like fill them with air and then spin them so that I can use them again, but I only want to put the side where my hands are on the side. Like I only want to put my hands on the inside or else I'll have just touched everything that I was trying to protect myself from. So that's always fun. But we have, what other cool things do I have up here? These are just more bacteria. I always think bacteria are cool. So, oops, I'm moving the wrong way. Yeah, so we've got some more dots. These are different pieces of DNA that I wanted to make and put in, okay, there we go. Put in another bacteria. So what these do is there's DNA inside these that I will pull out, put in these little tubes, and then be able to put in other bacteria, and it'll turn off certain genes. And what we'll see is whether or not the bacteria needs that gene to survive. If I turn it off and the cells are still alive, it means it didn't need it that much. If I turn off a gene and they die, that means the cells needed it. Does that make sense? Awesome. What other fun stuff do I have? These I just think are cute. Teeny tiny tubes. Look at how small that is. I think these are adorable. They're so small. They're like chiclets, like that candy. We have, let me find something that's not sterile. Here we go. So I have this nice rack, and this is just some, it's a chemical called EDTA. You don't need to worry about what it does. It's just, I wanted to show you something else. So remember how we saw those cool pipettes? These guys that let us pipette really small volumes? We also have ones that let us do big volumes, but really accurately. So full effect, we'll get the flame going again. Nice. You'll do is you rip it open, kind of like when they do a strep test or a strep culture at the doctor's office, and you put it on this thing that, if you can hear it, it sucks and spits out a liquid. So if I put it in this solution, I can suck up that amount that I want. So here, this is 5. It's hard to see at this angle, but there are little numbers on here, like a graduated cylinder, and then you can eject it back out. And you can use that if you want to take out a certain amount of something and you want to know what it is. What else do I have? We might as well. So the bacteria are on the plate, right? They're on this thing. And if I want to pick them up, what I do is I take these little sticks. Gotta keep them sterile, so I only open them when I have the flame on. And you'll notice they have a little loop on the end. 1 second and I'll put my hand under like a beauty YouTuber. That little loop on the end. And what I'll do is I'll open the plate with the bacteria on it and use the little loop to pick some up. Let's see. I don't know if you can see that I'm like spreading it around. So this is one of the differences between that E. coli and mycobacteria. You'll see that it's like really waxy, and I'm just kind of moving it around. It's like it's not all coming off the plate. Here you can see it's stuck to that loop a little. See that beige bit? Sorry, the focus is really bad. 1 sec. Da-da. So there's that little beigey thing on the loop. That's a bunch of bacteria. So this is a mycobacteria. So it was waxy, and I kind of just like spread it around when I tried to pick it up. But E. coli has a different cell wall, so it's a different texture when you go to pick it up. So let's grab this and another good loop. Grab one of these guys. So here, they look a little different. And then when I go to spread around one of these little circles, you'll see the whole thing comes up at once. It doesn't like move around as much as the others, and I'll use the other end to show you the same thing. So see how it comes all up at once? It's like creamy. Yeah, the E. coli is a creamy bacteria, and mycobacteria are waxy. So bacteria have all different types of like surfaces and properties, and that's one of the ways they survive in the body. So since they have that really hard, like that waxy outside, it protects them from the immune system. So I'm just checking the chat. One sec. Talking about lab exams. So yeah, when I was, not right now, so right now as a PhD student, I do research and I don't really get grades on it. It's just if I am learning and finding something, like more understanding how these bacteria do what they do. But when I was in high school and college, we did have lab exams. So basically they'd give us a task, like make an experiment to test if caffeine increases your heart rate. And we'd have to figure out how to make that experiment like perfectly. So you need to have these things called controls. Basically, you want to make sure that this is because of the caffeine. So maybe just drinking something increases your heart rate. So we'd also have like, oh, some people drank water. And you want to make sure you're measuring heart rate the right way. So we would do that experiment and then write it up in a formal lab report. And then they'd grade that both on how well we designed the experiment, did it, and wrote about it. Can the bacteria let go and leave the plate? So no, they really like what the plate is made of. It's kind of like if someone made you a bed out of pizza, you wouldn't want to leave it. You just kind of stay inside. Oh, we have a thumbs down for pizza. I'm out. No, pizza's great. And okay, we have a thumbs up for pizza as well. Good. So they don't just like lift off, which is quite good for us because if they were just going everywhere, I'd never get anything done. There are always accidents everywhere. I've accidentally like burned myself with the flame, just like singe my arm hair. Nothing that big. Just like if you're baking or something. But we do a lot of stuff to keep us safe. So the gloves, you'll notice that this is in like a little carousel thing so that you can't really knock it over. Yeah. And then when there's like a real pathogen, you'll be in one of those special safety hoods that I showed you before, that kind of thing. So Duncan asked about astatine. No, I don't know what that is. Let's Google it, or you can tell me in the chat, both work. It's a short-lived isotope? Oh yes, it's an element. Okay, yes, yep. Has almost never been seen. Interesting. Huh. Apparently it's the atomic number 85 and the rarest naturally occurring element in Earth's crust. Oh, it's because it only comes from heavier things. So you have to wait for something bigger to like break up a little bit to get that element. That's super cool. Thanks for telling me. Thanks for sharing. That's awesome. Yeah. Is there anything else you want to talk about? Have you tried using human DNA? So not here. We actually, one day when you can come back to Rockefeller and do like the in-person lab experience, some of the things we do is like extract our own DNA. So you can swab your cheek and then put that in a solution, a liquid, go through a whole experiment using different chemicals and things to pull out your DNA, and then you get what looks like a spitball, but it's all of your DNA. super cool. Rock EDU does that. So once the pandemic is over and we're all able to meet in person again, that's one of the things you can do here. There are other labs that use either human or eukaryotic DNA, but I don't. I think bacteria are cool enough, but that's just my preference. Yeah, anything else?
There's a question about how many people are in the lab right now.
Ah, thank you. I apologize for not seeing that. So right now, I think there are 3 or 4 of us. So there are about 15 people who are all part of our lab. But because of social distancing, we can't all be here at once. So we kind of break it up into shifts. Some people come in the morning, some people come in the evening, that kind of thing. So right now it's just a couple of us. And if we're not at the benches, science is also a lot of like writing. So we have desks where we do write-ups and we keep a notebook of all the things we're doing, that kind of stuff. So yeah, there's probably like 3 or 4 of us right now. What do you think is the coolest bacteria? Oh man, that's hard. I feel like I have to say tuberculosis because I study it, and probably because my boss is listening. But I'm not sure. Maybe some of the faster ones are cool. And some of the ones that talk to each other are really cool. Like bacteria can send signals to each other to say like, let's all grow at once. Or bacteria can make their own antibiotics to attack other species, other types of bacteria. So those are really cool. Those are things like Enterococcus bacteria and like Pseudomonas aeruginosa does that like talking to each other thing. Someone asked, is human DNA harder to work with? It depends. At the end of the day, it's the same set of A, T, C, and G. So the chemical, the molecule itself isn't any more difficult to handle. Usually it's just that you like need, there are special like rules when you're working with stuff from people. Just to make sure like you don't accidentally like share someone, like you don't want everyone to know everything about you. So we have some special rules for that, but no, same molecule at the end of the day, which is nice. Just taking a peek. Ah.
Nice. Any last questions before we wrap up? Feel free to unmute yourselves as well or continue using the chat. What do you want to be when you get older?
Me? Them? Yeah. Oh, right now I think I still want to do science, but ask me again in 2 years when I'm doing my actual like thesis stuff. Duncan wants to be a musician and environmentalist. That's a really good combination. You have something more artistic and creative, something a little bit more sciencey or policy, super cool. Someone asked, am I the only one in my family that studies science? No. So super interesting. I have an identical twin sister who is also a PhD student. She studies another type of bacteria at a different school. But no, my dad works for an office supply company, and my mom was an elementary school lunch aide. So no one else in my family does science. It just worked out that my twin and I both really like it.
Awesome, thank you. So if there aren't any more questions, we can wrap up. Thank you all so much for being great sports and asking such great questions and engaging with us today.
Thanks. It was awesome sharing this with you.
Thank you.
Awesome. Bye.
Bye.
Bye, everyone.